So let's talk about learning. You put a mouse in a maze and it navigates the maze quicker and quicker to find its food source. We refer to this as neurological learning. Now when you put an amoeba in a similar maze in a capillary tube, a little single-celled amoeba, it navigates the maze quicker and quicker to find its food source, just like the mouse. Amoebas don't have neurons, so how are they learning? To add one more example to this, a plant, a mimosa plant, and not the mimosa that you had for breakfast this morning, furls as you touch it. It curls up. But the more and more you touch it, the more it desensitizes to your touch and it stops furling. Then when you bring in a novel object, such as a gardening tool, it furls right up to it, and then 30 seconds later you touch it again with your human hand and it doesn't furl. It learned not to furl. Now the question is, plants and single-celled microbes don't have neurons. They don't have this capacity of learning that we hold so dearly to vertebrate systems. So this is why we have to look at microtubules. Microtubules were thought to be infrastructure, a road, if you will, that cargo would transport up and down its pathway, but nothing more. But as we dive deeper into microtubules, we find that they have a lot more physiological function than we thought. For instance, they have decorations, like the decorations on your Christmas tree. They have post-translational modifications, and these post-translational modifications encode information. They localize sequences, and they scaffold other proteins. Additionally, a microtubule propagates electrical currents. Now, this is a function that no other organelle has been shown to do. And this is why we need to look at microtubules and not neurons as the underlying mechanism for learning and encoding information.